Hi, everyone. My talk's a little different. I'm an environmental health scientist by training, but I'm Rosemarie de Rosa, and I'm at UC Berkeley. I'm an assistant professor there, and I also have a joint appointment at UCSF. So I just want to introduce my lab. I do research on stress and inequitable environments, and to frame that, I want to bring you to the social ecological model where we think of the individual, right? Their exposures are influenced by a number of cultural policy level factors that can really shape the number of resources that are available to an individual. And this interplays with a number of the individual level factors that can change behavior and influence the host level factors. And that interaction with the environment can change their individual biology and physical adaptation, can increase disease risk. And since we know that these exposures are inequitably distributed within a population, this can potentially contribute to the existence of health disparities. So what am I interested in? I'm really interested in how this social environment is biologically embedded over the life course. And I use tools from environmental toxicology and molecular epidemiology to really understand how social determinants really overlay with all of these fields. And so just to put this into context, we recently published on a study looking at, or on a theoretical framework of thinking about, we know, for example, that air pollution and psychosocial stressors are inequitably distributed in communities of color. And we're thinking about, well, how do those interact to then increase susceptibility to atopic and asthmatic diseases? And so we know that a lot of the research has been done on understanding how air pollution might directly impact disease risk. And so less work has been done in understanding how psychosocial stress may influence biology to then influence this disease susceptibility to also chemical exposures. And so my research is really trying to think about, well, what are these mechanisms where these two exposures may be interacting to increase susceptibility to disease in pediatric populations specifically, because if you think of life course, you want to think of where is my biggest impact for intervention, and the earlier you intervene, the more of an impact you're going to have on disease prevention. So this is a big question of how am I doing this? So right now, my lab is really focusing on high quality collection of pediatric samples, specifically in populations that have been historically underrepresented or excluded in biomedical research. And that has its challenges, right? So thinking about how do you do that while building trust and also just collecting high quality specimens, because right now a lot of the biomarker work that we've done in this field has not been representative. And so how do we work to increase representation in that field? I also work on developing novel methods to measure these stress biomarkers, because as much as this work has been done since the 1970s, it's still kind of unclear what are the best markers. So I'm using allostatic load and cellular aging as two models to think about how do you measure stress in pediatric populations. And with that, thank you.